All right, thanks everybody for joining our webinar on optimizing tertiary analysis. Elevate your data modeling with data studios on Sakera platform. My name's Rob Newman, and I'm Sakera's product manager lead, and I'm joined today by Rob Syme, our scientific support lead at Sakera. Before we begin, let's go over a few housekeeping items. As Rob mentioned earlier, all questions should be submitted in the Q&A box, and we'll go over those at the end of the presentation. Please participate in the opening poll that will be sent shortly, and also there's going to be an additional closing poll before the live Q&A. And lastly, we'll be sharing the webinar recording by email. All right. Let's wait a second while people complete the poll. While you're completing the poll, if you have any technical problems, please also post those in the chat. All right. Let's get started. Okay, for the agenda for today, for this webinar, first of all, I'm going to give a really short overview of the Sakera product suite. Many of you may know that we had the Boston Summit a few weeks ago, and we released a whole suite of new products. So I'm going to talk about that for a little bit. And then I'm going to be focusing in on Data Studios, which is now in public preview. And then I'm going to pass the mic to Rob Syme, and he's going to do a live demo showing you a valid Data Studios use case. And then finally, we'll switch back to me to go over some future Data Studios milestones that we're going to be tackling in the upcoming 12 months. And then we'll have some time for a live Q&A. Okay, at Sakera, our mission is to advance science for everyone through software. We're the home of the modern biotech stack, making scientific data analysis accessible at any scale. Many of you may know that we author a suite of open source software that includes Nextflow, and this is the industry standard for writing portable, reproducible, data-driven computational pipelines at scale. We also built MultiQC, which is for reporting and analytics, and this is our open source tool to simplify and streamline quality control analyses. And finally, we also create Wave. This is for dynamically managing workflow container creation and distribution. We also support the open science community by providing containers. This is a new release that happened at Boston, and this uses Wave to dynamically generate a container image based on the tools you've specified and stored in an image cache, which ensures reproducibility across workflow runs. We also just released Pipelines at the Boston Summit, which is a community-curated list of the highest quality open source workflows, complete with curated test data. We also provide a suite of documentation. This is our central resource for analysis development with Sakera tools, including how to establish your own centralized hub for managing and executing pipelines, and how to leverage the cloud to scale your data analysis requirements. We also provide a forum. This houses our community discussion boards to help new users, seasoned practitioners, and everyone in between to most effectively use our toolkit. And of course, we support NF Core. We support this foundationally. It's a global community effort to define and provide a free repository of best-in-class analysis pipelines built with Nextflow that adhere to rigorous best practices. Finally, and what we're going to talk a little bit more about today, is our end-to-end Cicera platform. This provides the one-stop shop for all of these tools and additional features for delivering modern bioinformatics at scale, and includes compute. This is our brand new, fully managed environment that prioritizes analysis initiation, data security, scalability, and privacy. And again, this was just announced at the Boston Summit and is now in private preview. We also provide Data Explorer, which allows you to securely browse and interact with your organization's remote data repositories, hosted in cloud storage buckets on AWS, Azure, and GCP, whether that's previewing files, selecting inputs for pipeline runs, or viewing results generated by pipeline runs. You can also download and upload files to your local host. Data Studios, which we're going to discuss in more detail today, and that was just announced at the Boston Summit to be in public preview. And finally, Fusion. This is our cloud-native, lightweight, and performant file system for data management in the cloud. And this underpins much of the platform functionality, including Data Studios. Okay, moving on to Data Studios. This enables users to securely spin up interactive analysis or development environments like Jupyter, RStudio, and VS Code, combined with your selected data, compute environments, and credentials already linked to the Cicera platform by simple point-and-click commands. This provides you with a seamless co-location of your data and analyses, with a much quicker transition from pipeline runs to launching interactive analysis environments. You don't need to provision and manage independent virtual machines with all the associated user and data permissions, complications, and overhead. You can perform your downstream, post-pipeline, tertiary analyses, whether that's exploratory data analysis, training machine learning models, or building interactive dashboards with tools like Shiny, Dash, or Streamlit. You can also easily troubleshoot and even author Nextflow pipelines in the VS Code IDE by connecting to your results and your work directories and rerunning pipelines. Environments are standardized based on version-controlled Docker template images using Conda packages for better reproducibility, versioning, and provenance. You can also easily share and collaborate directly in interactive environments using a single click. This is currently only for Jupyter environments and VS Code. We're working on RStudio licensing because real-time collaboration currently within that platform is a paid offering. Finally, access and control is all via existing Cicera platform credentials and roles. With the introduction of Data Studios, there's actually a new connect role specifically for Data Studios users, which allows you to connect to running Data Studio sessions. And if you actually want to manage Data Studio sessions, that requires the existing role of maintain or above. So if you have an admin or an owner role, you can also create and delete Data Studio sessions. As I mentioned, Data Studios is now in public preview, and it's enabled for every workspace in the Cicera cloud. It's going to be available for enterprise deployments in our upcoming 24.1 release at the end of this month. Okay, and with that, I'm going to jump to a quick demo. Well, let me just share a different window. Okay. Here we have Data Studios. If you have it enabled in your platform, you can see this Data Studios tab along the top. And here we show all of the running Data Studio sessions that I have in this workspace. So I have a Jupyter session, an RStudio session, and a VS Code session. You can see here the batch environment that this is running in. So here, these are all on AWS. And we have a little icon showing the environment. So here's Jupyter, RStudios, and VS Code. This is also showing the region of the compute environment and the author of the Data Studio session, which is me. We also have the status of the Data Studio. And here, all of these are running. But once you've created a Data Studio session, it's created in a stopped state, and then you can start it up by using these drop-down buttons in the kebab menu on the right-hand side. So as you can see, it shows my user and also other collaborative users who are also in the running Data Studio session. So there's 2 people or 2 users in the Jupyter session, only 1 in the RStudio session, and then there's 3 users in the VS Code session. So how do I create a new Data Studio? Super easy. You come up to the top right and hit Add a Data Studio. And here you choose your container template image. Right now, we only offer these 3, but we're going to expand these and offer others. You can define your Data Studio name. So I can type in name here, and there's an optional description. Then we can choose our compute environment. Here we've got one that I've already created, and then we can mount some data. So here I can attach, for example, like 1,000 genomes data. So we have a public bucket, which is the 1,000 genomes bucket. And you can see here it's displayed on the right-hand side, and I can mount that data. And that mounted data will now be available to the Data Studio session. And you can mount any number of buckets. I can define my CPUs and memory and hit add. And that's now created the Data Studio session. So you can see it here, Studio Webinar Demo. And like I said, we can start, we can view the details, or we can delete this because my role actually allows me to do that. We jump in and view the Data Studio. Here we see details of the Data Studio session, the environment, the description, the data that's mounted, but also we see these checkpoints. And these checkpoints represent session persistence. So every time a Data Studio session is stopped, a complete snapshot's taken and saved in the working directory of the compute environment as a named checkpoint. And as you can see, I've got 5 named checkpoints. A checkpoint includes all the intermediary work and data not explicitly written back to cloud storage, and the notebook configuration, including the environment setup, settings, configuration, and dependencies. From these checkpoints, you can either serially resume an existing Data Studio session, or you can start a Data Studio session from the kebab menu on the right-hand side. Here we can click Start as New Data Studio. Oh, let me just jump back. Checkpoints. Okay. So this results in lowered costs because you can start and stop on demand by preserving everything about your environment, fault tolerance, improved reproducibility, and portability of analyses for both individuals and collaborative teams. This also means that administrators can effectively manage costs. If users leave a Data Studio session running, costs can potentially needlessly escalate. Administrators can safely stop and clean up a running Data Studio, confident that the user analysis and the environment state won't be impacted. Checkpoints can also be renamed to custom values to reflect specific stages of analyses, and new Data Studio sessions created from historic checkpoints will reference the original parent Data Studio and checkpoint. So as you can see here, I've got these named checkpoints. I can go down to the kebab menu in the bottom right, and I can edit the checkpoint name, and I can say original checkpoint, and hit update, and now that's got a new custom checkpoint name. So let's say I wanted to create a new Data Studio from this initial exploratory data analysis session checkpoint. I can go over to the right and I can say start as a new Data Studio, and this could be new analyses. And here in the description, it tells me that this was started from this specific Data Studio session. And it also has metadata that tells me that studio session and also the checkpoint that it was created for. Again, it includes the same mounted data and the same CPUs and the memory, and I can just hit add. And now it shows this new analysis, which is essentially a fork of the prior environment. All right. Let's have a look at what these actually look like. So I'm connected to this Jupyter environment. So let's go and have a look at that environment. Ooh. Here's the environment. And here we have pandas and plotly that are libraries that are already included by default in the template. And we can see here we can do interactive analyses, and we can also import data, import libraries, and we can create graphs and do analyses. Additionally, here we have a RStudio environment, and we have here a Shiny app that I've got running. And let me actually stop sharing very quickly and share my whole desktop, because I want to actually show what this Shiny app looks like. So here we can just hit return. Oh, okay, there looks like there's a problem with that. So let me just jump into VS Code. Here's a running VS Code environment. And here I have the RNA-seq repository that's been created or cloned to this local host or this Data Studios environment. And I can actually go in and I can edit this in real time. Also, collaborators can come in and edit this as well. Okay, let's see. I think that's all I wanted to demonstrate. So let me just share my screen again. And I'm actually going to be passing the baton to Rob Syme, who's now going to walk us through a use case of image segmentation, model training using Data Studios. So Rob, I'm going to stop sharing and pass it over to you.
Perfect. Thanks, Rob. I hope you enjoyed the webinar. Excited for new features inside of the Secure platform. So Nextflow is this phenomenal batch workload orchestrator, but there's inevitably some occasions where you need a human in the loop to sort of transform or make sense of the data. For example, like as Rob outlined, when it comes to interactive analysis, you might want to take the output to a Nextflow run into an RStudio notebook, perhaps a Quattro notebook, to produce a report or conduct some sort of interactive analysis. Alternatively, you might want to take the outputs of one run and feed them into a second run, perhaps another Nextflow pipeline. And Data Studios is this feature which provides the opportunity to bridge this gap between Nextflow runs and bring a reproducible, containerized, and interactive compute environment to the data. So if your data is already in blob storage, pulling that onto a local machine can be sort of cumbersome and potentially error prone. And it makes much more sense to continue this sort of secure pattern of moving compute to the data. We can use the Fusion file system to mount that blob storage inside a running Data Studio container and essentially treat it like an infinite file system. The public preview of Data Studios, as Rob has just shown, includes support for VS Code, Jupyter Notebooks, and RStudio. And I think probably most of the audience here understands what those environments are doing. And as popular as useful as those environments are, we know that there will probably be many tools and tool sets and workflows that don't quite fit neatly into one of those boxes. And so to support other tools, upcoming versions of Data Studio will bring support for totally custom containers. So let's run through an example of how we might use a custom Data Studio to accomplish some research goal. There's a lot of really great spatial omics papers and work happening at the moment. So we've picked up a recent paper to use as our example. So the example I'm going to run through today will be using data from this excellent preprint just released last month. The authors used a technology from molecular cartography to spatially resolve and quantify transcripts in maize to better understand leaf development. And the specifics of this particular paper are not super critical. But what really is important, and I want to stress, is that we can use the Secure platform to roughly reproduce a small section of this paper. Like today in the webinar, we only have 20 minutes, so we certainly won't be replicating with any great accuracy the work done by Perico and the other authors. But I just want to sketch out the workflow inside the Secure platform to give you an idea about the capabilities. So very, very quick background to the paper. We have this small section of maize leaf tissue, and we have stained the cellulose walls shown here in blue, and the Davy stained the nuclei shown in magenta. And this molecular cartography system is an optical platform that allows one to spatially resolve single transcripts, which we can overlay on these two strains. Each of these yellow pixels that you can see on the screen now represents a single RNA transcript for which we have an XYZ position and also the transcript ID. Now, from here, we can follow a similar pattern to sort of group the transcripts. But we could sum, for example, the counts of all the transcript IDs over the whole image to determine the mean transcriptional profile of the slice. But we can turn this data into a single cell experiment by determining the boundary shown of each cell shown here in red, and then grouping the transcripts by those cellular ID. And then once you have these cellular IDs, you can do things like cluster the cells based on the transcriptional profile. But for our purposes today, we're just going to focus on the particular task of finding these cell boundaries, a really important and crucial part of this workflow. And this task is what we might call image segmentation. And looking at this small image, you might think, oh, that's not so bad. I could probably just draw by hand the segmentation around each of these cells. But the problem is, as you get into large data sets, the problem gets quickly overwhelming. We're not going to want to do this for every cell in this image, and we're not going to want to do it for every image in the data set. So we need a next flow pipeline. Fortunately for us, we have this NF-core Molcart pipeline. And this shows a really simple overview of the pipeline, but we're just going to focus on this cell pose track, a single tool in this pipeline. And we have some automated models for cell segmentation, but they don't perform very well. What we have are these images we're going to feed into the pipeline. These images flow through pre-processing and into cell segmentation before joining the transcript spots here on the right. And you'll notice the cell pose module can take a custom module as as input. And we can produce this by manual training. The pipeline also has a convenient sort of training subset path. So we can take our images to produce training, and then we can use those training subsets to produce a model. But there exists this gap in here where we need to inject some human intervention. And we need real eyes and real hands to do this manual training. And this is exactly what we're going to use Data Studios for. So this is what we're going to do in the demo. So basically, we're going to do 3 steps. The first step is to generate training data. We're going to run NF-core Molcart pipeline to generate the training data. And this step 2, we're going to take that input training data into a Data Studio to produce a custom model. And I'm going to save that model to blob storage. And then step 3, we're going to take that model and rerun the pipeline again, but instead this time including the model as input, and run the pipeline from go to woe. Of course, after that, you might want to take those outputs of the NF-core Molcart run and take it into another Data Studios for downstream analysis. We're not going to do that today in the interest of time, but let's give this a go. So step 1, we're going to generate the training data. And so this is just an ordinary NF-core run, and we're going to use the next Secure platform to run this pipeline. Inside of this workspace, we have the Data Explorer, so we can use this Data Explorer feature to browse the data we already have in blob storage. So under this Nextflow Summit bucket, I have Molcart input, and I have my TIFF files as images for each of those stains. So we're just going to use these first 2 images. So let's go back to the Launchpad, launch the NF-core Molcart pipeline. We're going to do step 1, generate training data. Great. So I have the parameters, and I'm just going to select those as normal. The only option I'm going to check is this create training subset, which is going to subsample our images and create a training subset that we can use to manually create a model. And the input, we're going to use the sample sheet, which points to the input files I showed you a second ago. And the outdir, we're going to leave unchanged. So when I'm happy with that, I can click launch. And that Nextflow will spin up an instance to run my pipeline. It's going to take a little bit of time for that to run. So here I ran one earlier. So when the pipeline runs, it produces these reports. I can see the multi-QC report. And this is our image. You might recognize it from the slides a second ago. And the training subset has pulled out these 4 regions for training. And these 4 images we're going to use to manually train our model. So those have been saved to blob storage as outputs of this pipeline. So we have this new feature, Data Studios. We're going to take those 4 images into a Data Studio and use manual training. If I view the Data Studio, you can see here that I've mounted some buckets and some paths within those buckets into my Data Studio. I've mounted the top level bucket, Nextflow Summit. So this is the bucket I'm using for the project. And I've also mounted, just for fun, some subdirectory inside of the 1,000 genomes bucket. To connect into that bucket, I can simply click connect. And here now I have a running Data Studio environment. This is sort of like a desktop. It has all the features that I would expect. I can browse the data. So here under workspace data, I have these 2 buckets and they look just like normal files on a file system. So I have this training subset bucket. So these are our 4 training subset images that we're going to use to manually train. So let's do that now. I'm going to use the cell pose tool, which I have installed inside of this environment. I can just load one of those images. So again, I'm just going to browse this data as if it was a local file system, but this is actually calling out and loading files from S3. So I'm going to run the basic model. And it's done an okay job. But there are some things where I suspect it could have done a better job. So for example, here, I think if I just select this, I can see that's actually not what I want. I'm going to delete, I can view these models and I can select. I can re-annotate these sections. And when I'm happy with those re-annotations, I can create a custom model. So I'm going to train a new model with these masks. I'm going to call the model name webinar model. When I'm happy, it's going to run the custom model training, and it's going to be running on a GPU. This instance happened to be a GPU instance, so I can quickly spin up and utilize those GPU capabilities of the instance. And it's going to take a little bit of time for me to run this. So we're now on step 2, we're training the model. When I'm happy with that, it will move on to the next image. Now, if you're watching this, you might have noticed that I'm not actually an expert in spatial transcriptomics. I don't actually know that much about generating these custom models. But fortunately for me, I do have a colleague who is a legitimate expert in training these models. So this might represent a situation where you as a bioinformatician would like to collaborate and lean on the strengths of your colleagues to do some training or to use the data that you've produced in some specialist way. And to share this data, I have 2 options. I could either point to the input data set on blob storage to my colleague, make sure that all the permissions were set up correctly so that they could download the data, work with it on a local machine, and then upload the results back to blob storage. But each of those steps introduces a potential misstep. Perhaps I don't have the permissions set up correctly, or perhaps my colleague doesn't download all of the input data, or there's something goes wrong in the upload. Instead of moving the data around, what I'm going to do instead is just going to grab this URL. I'm going to paste it in Slack and share it with my colleague. And what he's going to do is he's going to log into this data studio, this running data studio, and it's going to help me annotate the data. So because Florian, my colleague, has access to this workspace, he can actually log in and view this running data studio and interact with the data studio as if it was a local computer. So there's no need to move the data around. Instead, we move the compute to the data. So you can see here now Florian is doing those annotations, like I've got my hands off the keyboard. And Florian is already doing a much more professional job than I ever would of this image segmentation. While he's doing that, I just want to note that there was no need to shuttle data back and forth between blob storage and Fusion. All of these environments are stable and containerized and reproducible. I can provision the resources that are needed, be those high CPU, high memory, or GPU instances, no matter what machine I have locally. The credentials are managed by Secure Platform and then made available to the workspace. So all I need to do is make sure my colleagues have access to the workspace. And as Rob Newman just showed, we have this checkpointing of these Data Studio instances. So I can roll back to and save particular environments. And lastly, we have this wonderful shareable URL that I can just pass to my colleagues to help them log in and use the Data Studio environments. Great, the training is going well. Florian is now finished up. So while he's doing that, I'm going to switch back to the Secure Platform. Have a look inside of the Data Explorer, inside of Nextflow Summit. Inside of here, we have the model, or actually rather, we go to training subset, models. I can see here we have the webinar model, the model that Florian is training right now. So I can use this as input into a pipeline. So I'm just going to grab that path. I'm now moving on to step 3. We're going to apply that model to the data, the full data set. I can click launch. I'm going to use the CellPose algorithm. I'm going to use the same input data set. And under the CellPose custom model, I'm simply just going to paste in that model that we generated using our Data Studios instance. When I'm happy with those outputs and those parameters, I can click launch. And now we'll have a new Nextflow run here that is going to use the custom trained model that we produced inside of the Data Studio instance and apply that model to each of our images. Thanks, Florian. I'm going to take this back now. And let's say we would like to inspect the outputs of one of those runs. Actually, I have, as an example, a previous run. So I have a run here where we've applied the model to our input data set. And again, we have a multi-QC report, which describes the outputs of the run. This is a relatively small data set, so we've just applied it to one image. We have the total number of cells that were classified by our image, the cell area, and all those details. But if we want a more graphical view, we can log in back into the Data Studio instance and use a visualization tool like Napari. We can load a file. For example, we can load our images. We split our stack, and then finally we can load our model. We can view the results of our segmentation model. So you can see each of these regions is the classification results from the application of our custom trained model. So here we've used Nextflow to generate a training set. We've taken that training set into a data studio to generate a custom model. We've taken that model back into Nextflow and the secure platform to run a pipeline to apply that custom model, and then move back into data studios to quality control and sort of eyeball the results of those models in a graphical way. So this sort of workflow is just an example about how you might use data studios. Obviously, we want to make this as flexible as possible. Science and research generally use a lot of different tools and a lot of different sort of workflows. And we hope you'll find application in your particular workflow. We'd love to hear any feedback you might have about how this might be useful in your own work. With that, I'd like to pass it back to Rob Newman. I'm going to stop my screen share and pass it back to Rob. And we have a couple of questions and perhaps a survey to get through.
Great. Thanks, Rob. I believe I'm unmuted.
Yep, sound great.
Good. All right. Well, thanks, Rob. That was a fantastic real-world use case, providing real value, and I hope people really understand the power of data studios there, and especially this collaborative process with Florian. So, as I previously stated, Data Studios is currently in public preview, and it's available to Cyxtera Cloud users today. Looking forward to the future milestones over the next 12 months, the first thing that we have up and coming is the enterprise release of Data Studios. So if you are not using the Cyxtera Cloud, but you have your own enterprise deployment, this will allow you to do tertiary analysis in your own cloud infrastructure. It's going to be part of the 24.1 release at the end of this month. After that, we're going to be focused on adding support for custom containers. In the example that Rob walked through, that is a custom container. So, but right now, you're limited to 3 or 4 container template images that we provide. But if you have some specialized analyses that require specific packages, or if you need specific applications like CUDA, or if you're required to provide your own container from your organization's container registry, we're going to support that. The goal is to have that available for the Nextflow Summit in Barcelona at the end of October this year. That's why I've got a little Barcelona flag and the Nextflow icon in this custom containers item. This will also be our general availability release of Data Studios. So if you haven't registered for the Nextflow Summit in Barcelona, please do so. We're going to be having a whole bunch of announcements around Data Studios there. Immediately after that, we're going to focus on session management. And what does session management mean? That specifically means auto shutdown as a user-defined setting. And this will avoid runaway costs. So when you initially create your Data Studio session, you can say, hey, I want a lifespan of a maximum of 4 hours. So as you're approaching that 4 hours, if you've got up to get a coffee or you've walked away, there'll be an auto shutdown and a snapshot of the Data Studio session at that point. We're also going to provide an administrator-defined policy for organizations to be able to limit the number of Data Studios connections and also the number of users that can connect. We're also going to add resource labels to all the component parts of Data Studios to help with cost analyses. Then it's moving on to a real-time cost estimator. So you or your administrators or your IT team have a real idea of the cost per hour of your running Data Studio session. In the example that Rob showed, he was using GPUs, a GPU-enabled compute environment, which is much different than the standard compute environment that's just powered with CPUs. So their costs will be very different. And finally, we also want to provide the ability to securely publish interactive applications and dashboards and share that with your collaborators. Right now, you can join a Data Studio session and connect to that session, but we actually want to be able to publish dashboards. So whether your collaborators are your data science team who want to share with C-level stakeholders, or whether you have wet lab bench scientists who actually want to see the initial outputs of a run and take that information to then iterate on the work they're doing in the lab, this is going to be really helpful. Do please note that the delivery of some of these features may shift in priority and hence timeline. And we also have many more features that we want to implement. based on both our internal roadmap and also feedback from our users. So I do encourage you to take Data Studios for a test drive, really dig into it. And if there are features that you really think are missing, then we would love to hear from you. And that goes for both good and bad feedback. What we want to do is ensure that we're building Data Studios for what our community of scientists and bioinformaticians really needs. So before we go to the live Q&A, please take a moment to fill out the poll that will now appear on your screen. All right, we're close to, well, we're at 80% participation in the poll, which is fantastic. Love to hear your thoughts and yeah, love to chat to you further about this.
Well, well, we got our participants filling in that poll, Rob. Do you mind if we jump to some questions in the Q&A?
No, let's go for it.
Great. So the first one is, on what platforms do we currently support these data studios? For example, it looks like it was running on AWS. Does it run on Azure or GCP?
Currently, no, everything's running on AWS Batch. We do have a goal of getting or adding GCP support later this summer. So that is the next platform that we're going to support. But currently, it is just AWS.
Okay, great. Secondly, what happens if my data studio runs out of memory? For example, if I'm a bit too conservative in the resources that I supply to my data studio.
Yeah, it will shut down. But when that does occur, we have the ability of doing snapshots before it is shut down. So this does happen. So let's say you're pulling in some large data files and you're trying to do analyses or sort of manipulation on the files in pandas, for example. There is a potential for it to run out of memory, but as I said, it will take a snapshot before it then shuts down.
Right. I have a question here. Does it support genomics applications like IGV?
Yeah, that's a great question. So when you fire up a Data Studio session, you do have root user permissions. So you can actually go and install any software now in the Data Studio session, and that is preserved when a snapshot is taken. So any specific custom software that you install is preserved. Now for graphical user interfaces, that's a little bit more tricky. Right now, we don't have the ability to support that, but as you saw with the demo that Rob just walked through, we are going to be providing an X11 template image in the next couple of weeks. So if you do have a graphical user interface like IGV, you can then create your own Data Studio session from that template image of X11, and then you will be able to have access to GUI applications like IGV.
It's a great point. So Florian and I had great fun. That X11 sort of general purpose Ubuntu container. We installed a whole bunch of stuff in there. We had great fun. You can install whatever you want, and so we found reasons we end up running an IGV in it, but you can really, the sky's the limit there. And we definitely like at Secure appreciate that like a lot of research contexts are very varied and very particular, and we don't necessarily want to impose limits on what sort of software you can run. So flexibility is key.
Yeah, and when we do deliver or provide this custom container, I mean, there are many customers who potentially have very specific version controlled and managed within their own container registries. Like there's very strict permissions, very strict limits around what applications can be installed. So we do want to be able to support that. So yeah, this bringing your own custom container whether that's taking our base image and augmenting it with Wave, if you just want to install a Conda package that is missing from that template image, or if you want to have your own, like I said, Docker file that actually lists all of the applications that you want, the plan is to support that. It is a little bit more complicated because we do have specific pieces of infrastructure that are required to enable data studios, but we're going to have very comprehensive documentation to ensure that you do add those component pieces so it does work natively within the platform.
Right. A question, and invite anyone to continue to submit questions to the Q&A inside of the Zoom chat if you have particular questions you'd like us to address. I have a question here. Where is the state, when you shut down a Data Studio instance, where is that state saved?
Yeah, that's a great question. In the compute environment that you have associated with the Data Studio session, so as I showed, every time you create a Data Studio session, you have to choose the compute environment. Whatever you define as the working directory of the compute environment, that's where all the checkpoints are stored. So if you just have it at the bucket level, there will be a .studios directory, and within that directory are all the checkpoints associated with all the Data Studio sessions that are running or have run in that compute environment.
Fantastic. Oh, and a great question here. If I would like to share Data Studio with a colleague or collaborator, they need their own AWS account?
They don't necessarily need their own AWS account. They need to have credentials associated with their user in the Sakera platform. That's all they need. Everything is handled in the platform side, like that's all abstracted away in the credentials and the permissions around the compute environment. So provided that user has the connect role or above. So we have many roles within the Sakera platform, the viewer role, the launch role. We have this new connect role. The launch role and the connect role and above, all of those user roles in the workspace have the ability to connect to a running Data Studio session. So provided your user has that role associated with them, then they can collaborate in real time within the Data Studio session that you've created.
Wonderful. Very nice. I think that's most of the questions. Last chance to get any questions you might have into the Q&A box. Failing that, I think, are there any closing remarks? I think that will probably wrap up our Q&A. Unless there's any other questions you have, Rob.
Yeah, no, I think that's it. I think, I just want to thank everybody for their attention. Data Studios is new. We're seeing a lot of excitement around it, so we're really excited about it. And yeah, any questions, any concerns, if you want a demo, then please reach out to us. And it's under active development. We're rolling out new features all the time. And if you want to keep abreast of those, just follow the Sakera blog, because we're posting, as we release new features, blog posts about it.
Perfect. Thanks all. Thanks all for attending.
Yeah, awesome. Great to chat to you all. And please reach out if you need further information.